You told me an interesting story when we last spoke, Sanjay, about the way that your department, specifically at AbbVie, interfaces with the greater manufacturing environment there, sort of the org structure. Can you kind of share that with us a little bit? So tell us how the work that you're doing is applied across the manufacturing environment there.
So we are part of process development. We are part of global process development. We identify new technologies. We work with manufacturing, supply chain, to see how the new facilities we are putting up, or new manufacturing facilities we are putting up, or how existing facilities we have, how we could develop a technology such as PAT or intensification technologies for them. So when we develop a new process, we'll integrate those into our facilities much more seamlessly. Yeah. That's where we are at the moment. We identified a couple of technologies. We'll test in the lab first. We'll look at the robustness with this intensified process. Then we transfer to our pilot lab. We got very good collaborations with pilot lab. And then we'll identify some of the molecules where we could implement those going forward. We take a stepwise approach also. We identify some technologies and do a stepwise approach based on the risk assessment and the needs for the projects we have.
Irina, what about at AstraZeneca? Is your capacity sort of designated in a specific process, or similar to Sanjay's role at AbbVie, are you applying learnings from your work across the manufacturing environment there?
Both apply. So capacity is linked to which molecules we envision for a particular landscape in the next few years. Readiness, it's almost like we build these roadmaps that they will eventually converge. Readiness of technology, the regulatory strategy, the quality assurance around all of that. Critical decisions on readiness of the portfolio projects. Is it before the final approval, or it will be more on an LCM, or life cycle management? And of course, the forecast of the need for that particular drug, what we expect, and how many of those in parallel would be operating. From a process development perspective, so my group sits in the middle of biopharmaceutical development, which means we have to work with pretty much every single function to make these work, while we bring novel technologies. In order to implement, we have to have very strong stakeholder management. So we bring the current, and that's how we started. We adapted the current purification process platform. Of course, the upstream process was developed for higher productivity in a fusion bioreactor. And then we create tools to fit the process to this new mode of operation, which is a lot more complex. And when we bring these tools to light, as we transfer technology to our partners and our stakeholders, we will be testing the technology in a pilot scale first. Envision how different would that be when we scale that up. And scaling that up, is it clinical versus commercial? How many projects? How many will have to be operated in parallel? Sanjay highlighted the difficulty and complexity of the amount of buffers that need to be produced. So those are even different groups that now their operation changes from running batches in parallel with the continuous or perfusion bioreactors with the entire operation. And I want to highlight, it is super important that we test our mode of operation in showing and proving that we have a closed system operation. Because the levels of course, contamination or bioburden that are acceptable, the bioreactors, they zero. For downstream, we could operate at low bioburden, but in this particular mode, we don't want to operate at all with those risks, because it's a 24/7 operation for a few weeks. So all of these needs to be conceptualized and tested as a proof of concept, and bring it to light, and then through engineering runs in the GMP facility.